So in the previous chapter on metabolism, we looked at ATP and energy coupling. In this chapter, we are going to see how the food we ingest is converted into different molecules and in that process also makes ATP, utilizing oxygen that we breathe in. This respiration process does not happen in a single step. Like the equation show that carbohydrate plus oxygen will release carbon dioxide and water, it happens in multiple stages. So at the beginning of this chapter, we are going to focus on the basic concepts of how ATP is made and what are the different electron carriers used to excite the electron and then carry it to one step from another step to the oxidative phosphorylation that happens at the end to make the most amount of ATP. So this chapter is also one of the most detailed, complex chapter you need to understand and organize them by sections. So the first part of this respiration process starts with glycolysis. So glyco refers to the sugar, lysis means breakdown of the sugar. So the glycolysis is where a carbohydrate such as a glucose is broken down into 2,3 carbon molecule, and in that process you also extract high energy electron and give it to the electron carrier NAD+ to make NADH. Once you make that NADH, that transfers the electron inside mitochondria and communicates to the enzymes there to shunt it to oxidative phosphorylation step. But in this process, it also makes some ATP as substrate level phosphorylation. So that is not sufficient for the cell to have all the functions done. So the energy is also carried on by the two small 3 carbon molecule transferred into the mitochondrion, which are converted into acetyl-CoA. That is the second step of the aerobic respiration process called acetyl-CoA formation. But the acetyl-CoA can also come from fats through a process called as beta oxidation process. So that process also makes acetyl-CoA, and acetyl-CoA formation makes NADH and also releases some carbon dioxide that we breathe out. The acetyl-CoA enters the next pathway, we call a cyclic pathway called Krebs cycle, the first classical cyclic biochemical pathway. It is also referred to as citric acid cycle. There, acetyl-CoA is oxidized, means electrons are extracted and given to the electron carriers NAD+ and FAD to make NADH and FADH2, which then go on to oxidative phosphorylation. There is also some ATP made, but not a lot. Okay? Just a few ATP there, and then the NADH and FADH2 made in glycolysis, acetyl-CoA formation, Krebs cycle, all of them combined together go to oxidative phosphorylation. So oxidative phosphorylation happens on the inner membrane of the mitochondrion, and the NADH, FADH2 gives all the electrons that they extracted from the high energy molecules that we consume as food, and that electron transport happening in the electron transport chain allows protons to be pumped inside the intermembrane space of mitochondria. So when it creates a gradient of high proton concentration in the intermembrane space, low proton concentration in the matrix, the protons come back through a process called as chemiosmosis. That is what makes a lot of ATP by the enzyme ATP synthase located on the inner membrane of mitochondria. So the oxidative phosphorylation is a process where the oxygen that we inhale is utilized to accept the electrons at the end of this electron transport chain. But the carbon dioxide has been released already from acetyl-CoA formation step as well as in Krebs cycle. So in this process, the electrons coming at the high energy level are dropped down in energy level, and oxygen is the one that accepts the electron at the lowest energy level, converts into water. Okay? But when we run out of oxygen, all we got is 3 minutes to survive. So how does the body cope up with that? When the oxygen is deprived of the cell, the electrons are not removed from NADH, NAD+ is not regenerated, FAD is not regenerated, glycolysis comes to a grinding halt, Krebs cycle and acetyl-CoA also stop. So to circumvent that, fermentation happens. So the NADH made in glycolysis is utilized to convert the pyruvate to a lactate in lactate fermentation, or the pyruvate is converted into acetaldehyde in alcohol fermentation, and the acetaldehyde is converted to ethanol. So the reason for fermentation process is for the lack of oxygen, the cell needs to survive by making ATP. Without ATP, there's no life. There's no cell cannot survive that. So that is the process of fermentation we will cover at last. So when you study this chapter, focus on individual pathways: glycolysis, acetyl-CoA formation, Krebs cycle, oxidative phosphorylation, and fermentation. Then you look at what are the net inputs that go into the pathway, what are the outputs that come out of the pathway, and lastly, you want to focus on what regulates this pathway, what is the key regulatory enzyme, and how it is regulated. Don't worry about memorizing all 10 steps of glycolysis or all 8 steps of Krebs cycle and so on. Try to get the big picture and then focus in on bottom line, inputs, outputs, and the key regulatory step. That is the respiration chapter.